Hello. In the previous video, we saw how cells divide by mitosis whenever they're trying to make identical copies of themselves. Today, we're going to be talking about sexual reproduction and a different form of cell division that we call meiosis. Inheritance is the process by which the genetic information is passed from the parent to the offspring, because we all began as a fertilized egg. That egg had all of the information from both of your parents that then is going to undergo trillions of divisions to form a multicellular organism. So how does this happen? Sexual reproduction is the process by which this happens, and it is an evolutionary advantage. Remember, when we talk about evolution, we define it as the reproductive advantage of individuals that are better adapted. This is one of the advantages, because we're going to see in sexual reproduction, we're going to shuffle information, so that leads to variation among the offspring. You look very similar to both your parents, you may look similar to your siblings, but everybody is different. Okay? If we reproduce by mitosis, everybody will have exactly the same DNA. So that's not advantageous, because if something comes that affects a particular genome, it will wipe up everybody. Remember COVID, hasn't been that long since COVID. There was people who got very, very sick and had to be put in respirators. There was people who were suffering like a mild cold. And there was people like me. I assume I had COVID. I was teaching during those days, but I never had any symptoms. Why do different people react different? Because we're different. Okay? So variation allows for some individuals to die, but others survive. And that variation comes from sexual reproduction. And since most eukaryotes reproduce sexually, that takes care of this problem. But mitosis should not do that. Mitosis should not produce gametes with only half the number of chromosomes. Mitosis should not produce variation. So we have a different type of cell division that we call meiosis. So you divide the DNA of your diploid cell into two haploid cells that we call gametes. Why do we have to cut down the amount of DNA? Because think about humans. Humans have 46 chromosomes. If the egg has 46 and the sperm has 46, what happened to the zygote? It will have 92 chromosomes. So obviously this won't work, because the number of chromosomes remains constant for the species. Now in meiosis, we make a haploid sperm, meaning it has only 23 chromosomes. The egg is haploid too, only 23 chromosomes. So now the zygote has 46, so you regenerate the correct number of chromosomes. Obviously, you cannot pick up any 23, but remember, we have two copies of each chromosome. So what the cell does is gives a full set to the gamete in here. So right there, you can just say that if you're combining chromosomes from the sperm and from the egg at fertilization, that introduces variation. But we are going to see that there's a whole lot more to that in meiosis. And that's what explains that you don't have two people identical. Okay? And the only identical people, identical twins, if you remember, those are twins that they began as one zygote, and at some time in early development, it is split into two, and the two of them continue developing, but they began as one zygote, so that's why they have the same DNA. Now, so we talk about, depending on where you have mitosis or meiosis, you can have diploid or haploid organisms. We're not going to get into too much detail here. We'll see a whole lot more of this in the second semester. But in animals, usually the big organism is diploid. Okay? Now the diploid is going to produce haploid gametes. Again, think on the person, your body's diploid, your gametes are haploid, so you're much bigger than an egg of an sperm. So we call animals diploid dominant. Fungi, such as mold, mushrooms, something like that, the biggest organism is actually haploid. They're haploid dominant. And only a very small part of the organism is actually diploid. And once again, I'm not going to waste time on this one because you'll cover it in a lot of detail in the second semester. Plants are more interesting because they alternate. You have a multicellular haploid and a multicellular diploid, and you're going to see the alternation of generations. Again, we'll see all of that in the second semester. So how do we make haploid gametes? Through meiosis. That cuts down the number of chromosomes, and we're going to see that it shuffles the genes. So in addition to combining genes from two different organisms, we are going to shuffle the genes within each of the parents. Okay? So it all begins first with the normal interphase, because you need to duplicate your DNA. And now you go through two different divisions. Okay? You have meiosis I and meiosis II. We'll see that sometimes there is a very short interface between them, sometimes there isn't. We'll get to that. But meiosis I is the one that reduces the number of chromosomes in half. So we call that the reduction division. It's going to have similar stages to mitosis, so we call them Prophase I, Metaphase I, Anaphase I, Telophase I. Meiosis II, on the other hand, is going to be very similar to mitosis, and the stages are going to be Prophase II, Metaphase II, Anaphase II, and Telophase II. You see that the interesting stuff is going to happen in the first division, so we are going to be looking at these phases in a little bit more of detail. Okay? Remember we talked about homologous chromosomes, the matched pairs that contain genes for the same traits. Okay? During Prophase I, homologous chromosomes are going to line up together. That process is called synapsis. You have the two chromosomes that you can tell they have different traits, different variations of the traits, but they're identical in shape and everything else. When they come together, actually, they put them separated here for you to see it very easily, but they usually sit one on top of each other, and some people call that a tetrad, because you actually have 1, 2, 3, 4 sister chromatids in there. But regardless of how they get together, you see that some of these chromatids are very close to each other. If one gets on top of each other, sometimes these bonds break and they just swap pieces. Okay? This is called crossing over, and that happens between non-sister chromatids, meaning the sister chromatid of one chromosome, the blue, exchanging information with a sister chromatid from the red. So now you have a chromosome that's mostly blue with a little red piece, or a red with a little blue piece. This is very important. Let me give you an example. Let's say that the blue chromosome calls for black hair, and the red is going to be blonde, and the black hair is going to have black eyes, but the blonde is going to have blue eyes. So you say, oh, all of the children are going to have either black hair, black eyes, or blonde hair, blue eyes. But when recombination takes place, yes, you still have the black hair, but now you put the blue eyes in here. And this one was the blonde, but we just move the piece in here that calls for the black eyes. So now you have children who have black hair and blue eyes, or they're blonde with black eyes. You see how you're increasing diversity? And of course, crossing over can take place more than once, but once again, you're mixing the genetic information. Okay? You exchange genetic material. These new chromosomes that have some maternal, some paternal DNA, we call them recombinant chromosomes, to differentiate from the original ones that were all from the mother or from the father. During Metaphase I, just like in Metaphase, homologous chromosomes are going to be arranged in the center of the cell. And now remember, in mitosis, the chromosomes line up. Here you don't have chromosomes, they're the pairs of chromosomes. You can see in there in the drawing that you have pairs of chromosomes lined up. You can see it in there. You don't have all of the chromosomes in one line, you have pairs of chromosomes. Now, the orientation is random, meaning you're not going to have all of the chromosomes for the father on one side and all of the chromosomes for the mother on the other side. So when they separate in the next phase, all of the paternal DNA goes one way, all of the maternal. Rather, it's random. So you see the different possibilities, because in here you're having both blues on one side, both reds on one side, but in this one we have a blue and a red and a red and a blue. So when they separate, they're going to form, in this particular case, they're going to be all blue or all red, but this one is going to have blue and red, red and blue, and of course, in the second division, the chromatids separate, so every chromatid, every resulting cell is going to be different, has a different chromatid, because of this shuffling in here and there. So we call that independent assortment. And by the way, the number of possible gene combinations is 2 to the n. In this case, we were talking about 2 pairs, so there's 4 combinations. But you get an idea when you talk about humans, that you have 23 pairs, then the number starts becoming, not only the DNA has already been shuffled, it will be crossing over, but now we're going to shuffle much more during independent assortment. And of course, then you combine that with fertilization, you may have only one egg, but you may have several million sperm, probably all of them different. So that's why the chances of getting 2 individuals that are identical are close to 0. During anaphase I, now you don't separate the chromatids, you separate the homologous pairs, meaning one homologous goes one way, the whole chromosome, okay? Since the sister chromatids remain tightly bound at the centromere, you're moving chromosomes instead of chromatids. But because the chromosomes have arranged randomly during metaphase I, some of the chromosomes are going to be paternal, some are going to be maternal, but they're moving in opposite directions. So you see in there that you have 2 red and 2 mostly red and 1 mostly blue going one direction, 2 mostly blue, 1 mostly red moving in the opposite direction. Telophase I, that means that the separate chromosomes have arrived to the different poles. Whether you reform the nucleus or not depends on the species, okay? But you're going to have cytokinesis because you want to divide the cell. The 2 cells that you have are haploid. They have half the number of chromosomes because we just split the chromosomes in half. And another important thing, sister chromatids are no longer identical. Before they were identical, because you remember sister chromatid was made up of replication of the first chromatid. But now with the crossing over, you don't have identical sister chromatids. So you cut down the number of chromosomes in half, and you have different sister chromatids. Now, some cells have a brief interphase, they take a little break. It's called interkinesis. The only thing is that if you do have interkinesis, and like I said, depends on the particular cell, there's no S phase, because we are trying to divide the DNA. So you don't want to duplicate the chromosomes when you're trying to cut down the number. Makes sense. Okay? Some cells don't have interkinesis. Okay? But meiosis 2 is going to be very similar to mitosis. Anaphase 2, whether we can prophase, metaphase, the chromosomes line up just like in mitosis. Anaphase, the chromatids. Now, in anaphase 2, the sister chromatids are separated. Remember, anaphase 1, it was the chromosomes. Now it's chromatids again, just like in mitosis. And then telophase, if you have 2 cells and each one divides in 2, you end up with 4 gametes. They both are haploid, because these original cells here are haploid, and because you separate, they have only 1 chromatid, and each one is different. You have 4 possible gametes, and each one is different because of the combination of crossing over and independent assortment. Okay, and like I said, you still increase diversity more with fertilization. So this is just putting mitosis and meiosis side by side. Basically, mitosis, you begin with 1 cell that's diploid, and you end up with two cells, they're diploid, and they're identical. Okay, you can review if you want to, but meiosis, you begin with one cell that's diploid, it splits into two cells that are haploid, and then it's divided in two, so you end up with four cells that are haploid, and they are different from each other. Okay? So, big difference in the type of cell division. Remember, we use mitosis when you want more cells of the same for growth, for repair. You use meiosis when you're trying to increase variability, when you're making gametes for sexual reproduction. Okay? When people started studying chromosomes, I still remember I was an undergraduate. You took a picture under the microscope during mitosis. Usually Prophase or anaphase were good ones. And we actually printed the picture and with a pair of scissors cut down the chromosomes. A karyotype shows you the number of appearance of all the chromosomes. It shows you which one is bigger. The binding pattern, you're going to be seeing in different colors. You use different dyes, but those are dyes that bind to specific DNA sequences. So you can tell when you have the same DNA sequence in both of them, where is the centromere located? Is it in the center, or in some of them it's going to be located more towards one end than the other? And of course, it tells you male or female. In this particular case, there's an X and a Y chromosome, so that's a male. If there was two X chromosomes, would be a female. Okay, and you can study lots of genetic problems this way. Fortunately, today you don't have to cut them with scissors. Computers will do all of that work for you. Sometimes things go wrong with meiosis. Okay, a very common problem is non-disjunction, meaning the homologous chromosomes or the sister chromatids fail to separate. If it's meiosis 1, meiosis is represented with an R, okay, and an exclamation. So during meiosis 1, sometimes the homologous chromosomes don't separate, leading to different number of homologous chromosomes in the resulting cells. So then, even though they undergo meiosis 2, you end up with the wrong number of chromosomes in there. Sometimes meiosis 1 takes place fine, but the sister chromatids fail to separate. This would be during meiosis 2. But either way, when something goes wrong, you end up with the wrong number of chromosomes in the gametes. Having the wrong number of chromosomes is called aneuploidy. And the most common problems are monosomy, when you lose one chromosome. So after fertilization, you should have two chromosomes, but you're missing one, so you have only one. Trisomy means that after fertilization, if one of the gametes has two chromosomes and combined with another gamete has one, you have an extra chromosome in there, 3, that's where. And sometimes it's not just one chromosome. Sometimes it's a complete set of homologous chromosomes. When you have more than the correct number, if instead of having 2 N chromosomes, you have 3 N, we call that polyploidy. And when we study plants, we're going to see that's very common in plants, and actually it does have a purpose. Okay? But these are the problems that are likely to occur. And a typical example is Down syndrome, trisomy 21, meaning that something goes wrong and you have 3 copies of chromosome 21. And everyone's familiar how a person with Down syndrome looks like, the problems that they have, and they tend to have an open hole between the 2 atria, which many times needs to be surgically repaired. They tend to have different degrees of mental retardation. There's a number of effects of trisomy 21. And everybody knows that happens when the mother is old. You can see the incidence of trisomy with the mother's age, and by the time the mother gets in the 30s, it starts getting iffy. If the mother reaches 40, the chances of trisomy 21 increases dramatically. And some recent studies have shown that, to a lesser extent, but it also happens with the father, that the chance of non-disjunction in the formation of the sperm also increases with the age of the father, but it's less common than with the mother. Something very interesting, and you have probably seen this if you go to the Olive Garden, where they have the little thing that you pay, and it has games. One of the questions that I've been watching for years is to say, a calico cat is, and they have 4 different choices. One of the choices is almost always a female. Well, I hate the almost because they are females. Okay. What do I mean by X chromosome inactivation? Well, we have a problem because females have 2 X chromosomes. Males have 1. So when you have genes that are on the X chromosome, does it mean the female gets twice as much the dose? It produces twice as much of the proteins called by these genes? Because then either the male doesn't have enough or the female gets too much. In order to function properly, 1 X chromosome in the female inactivates, condenses again, and it's called a Barr body. When you're looking in the cell under the microscope, instead of seeing a little chromosome, you see a clump. That's the Barr body. That way you only have 1 working X chromosome, and you don't get twice the product from that X chromosome. Okay. So which chromosome gets inactivated? That's random. Okay. You're talking about a particular pair, but one cell decides to inactivate the one that comes from the mother, now one inactivates the one that comes from the father. It's completely random. This is very useful whenever you have someone with an abnormal number of X chromosomes. There are people that have 3 X chromosomes. Well, you only inactivate 1. So you're still going to have some effects, but it's going to be much milder because you only have double the dose instead of triple. But going back to the Olive Garden, why do calico cats are females? Because the calico code comes from X chromosome inactivation. You have 1 chromosome that's responsible for the white color. Okay? And well, let me say 1 gene. And 1 gene on the X chromosome that you have a choice of brown or black. You cannot express both of them. Okay? So since the chromosomes, X chromosomes are randomly inactivated, some cells are going to express the black, some cells are going to express the brown, and that's why you get the particular pattern of a calico cat. But if it was a male, it would be either black or brown. You not have, because you wouldn't have 2 X chromosomes, 1 to inactivate. So calico cats are females. Next time you go to Olive Garden, remember. If all of these did not include enough diversity, you also have structural rearrangements of the chromosomes. Okay? These are some of the ones that most common that have been observed. You can have deletions. All of a sudden, part of a chromosome is lost. You have in there this brown part, it disappears, and this reattaches itself. You're going to see chromosomes break and reattach very, very easily in the cell. So you just lost some information. How serious is that? Depends on what you lost, obviously. Is that valid information, or is that something that's not very important? You have duplications, where 1 chromosome all of a sudden makes 1 part of the chromosomes duplicates itself and attaches it into the chromosome. So now you get twice as much. And some people have claimed that probably that's how evolution took place, that there was a series of duplications and rearrangements and things like that, and they led from a particular protein in an animal to the same protein in humans and all of that. We'll talk about all of that when we talk about evolution. But that is a fact that has been observed. A section of a chromosome is copied, and not just once, but several times sometimes. And in inversion, this is getting even more interesting. A part of a chromosome separates itself, flips upside down, and reattaches itself. So now what is being coded is different because it's like I get a word in here, like for instance the word added. If you reverse the order, it says D, E, D, D, A. Data. What's that? This particular case is nonsense. I know what added is, but data is nothing. Sometimes it may be that inversion does have a meaning. So it depends on what's going on. But basically, the inversion means a chromosome flips upside down. We talk about insertions, but we're talking there between chromosomes, okay? You can say, oh, every time this happens, like this chromosome, when it makes a copy, it gets inserted, but it's in the same chromosome. But sometimes you take a piece from one chromosome and you insert it into another chromosome. Okay, so that's between two chromosomes, interchromosomal. And this is important because you still have, hopefully, that gene intact, but by putting it in a different location, in the original location, there was always before that genes that control the expression of this gene. They said, this is when you're going to work, this is when you're not going to be expressed, blah, blah. When you move it to another chromosome, it loses that. It may never work because you don't have anything turn it on. It may be working continuously, so you lose control of the expression of this gene. And now you have translocation when chromosome segment is moved to another chromosome. Seems very similar to this particular, but if you notice, there is like a swap. It's not insertion because you have part of the green chromosome attaching itself to the brown, but part of the brown attaching. That's what happened during crossing over. Okay? So once again, you're moving information around. Best case scenario, you move whole genes, but you lose control over gene expression. Sometimes it may work out where you cut in the middle of a gene, and then obviously it will not work. So there's a lot of possible variations in here. So yes, I understand it's confusing, and I do have a table that I made where you put the main information about mitosis and meiosis, similar to what I did for photosynthesis and cellular respiration, where you can compare the two of them. It makes it easier to understand, easier to study, and of course, I'll be glad to answer any questions. So go to the video description, make an appointment in my Wycent account, and I'll be glad to share that worksheet with you and answer any questions.